welcome back, everyone. Give folks another minute or two to get settled. In this session, we're going to switch gears a little bit from some of the theoretical framework that we heard about in the earlier session to developing new theoretical constructs as well as computational mechanisms to handle some large-scale real-world data. The first talk in this session, David Heckerman, who's a distinguished scientist at Microsoft Research, is going to talk about new computational mechanisms and the associated theoretical frameworks to examine genome-wide association studies. But with that, I'll let David.
Great. Thank you, Sue, and thanks for the rest of the organizers for allowing me this chance to speak. And speaking of organization, I think, Sue, you asked me about 1.5 years ago to do this, so you guys get an A+ for planning ahead. Thank you very much. So let's see, Mike talked about big data, and Yuta and Thomas talked about causality. I'm going to attempt to talk about both. It's very difficult. The intersection is very difficult to work with. And in a sense, I'm going to cheat a bit. When we get to the actual problem, when I show you the problem in genomics that we're going to talk about, you'll see that the causal issues are very simple. Asking the question, does A cause B or does B cause A? That's very simple in this problem. So most of what we're going to talk about has to do with the statistical and computational issues around big data. But at least you'll see a little mix of both here. So again, we're going to consider a problem from genomics. The causality issues are fairly easy. The statistical problems will come from having lots and lots of dimensions. And I'll go into that in a lot of detail. And also, there's a lot of confounders that are present. On the computational side, the issue is going to come from sample size. So let's look at this problem and then get into the details. When you see a talk on genomics, almost always the talk starts with this slide because it's so important. I imagine many of you have seen it already. This is a plot of the cost of sequencing a human genome, the whole genome, as a function of time. So back in 2001, 2 teams did this for the first time. One team, I think, it cost them several billions of dollars to do the task. Another team, it was only several hundreds of millions of dollars to do the task. But since then, the cost has been dropping very quickly, faster than Moore's law, which you see on this curve. So today, actually this curve stops around 2013, but today, 2015, you can sequence an entire human genome for about $1,000. And not only has the cost been going down very rapidly, but the time it's taken to sequence a genome has dropped very rapidly. A decade ago it was years. Now you can do it in about a day. So this phenomenon, this trend, has led to a revolution in the production of genomic data. There's just a flood of data, and from that data are coming all sorts of interesting insights and all sorts of interesting applications. One of each I'll talk about right now. So in terms of insights, one of a very surprising finding came when we looked at all these human genomes and realized that if you take any 2 individuals, they only differ in their genome by about 0.1%. So each of us has 3 billion base pairs, A's, C's, T's, and G's. That's our genome, times 2 because we have 2 copies of each chromosome. But when you take the genomes of 2 individuals and compare them, you only see millions of differences. Most of those differences are single changes in a nucleotide from one individual to another. We call those SNPs, single nucleotide polymorphisms, or sometimes they're called single nucleotide variants. And then there's other things. There's insertions. Someone has a base pair where someone else doesn't, a deletion, or you can see copy number variations. Various parts of the genome are copied, and then how many copies you have differ from one individual to the next. For this talk today, I'm just going to focus on the very simple case of SNPs. But I want you to know that there's other things that are going on here. Okay, so that's one interesting insight that we've gotten from this genomics revolution. And perhaps the most important, most pursued application is personalized medicine, or sometimes called precision medicine. And I'm about to show you an example of that, but it's pretty gruesome on the slide. So if you're squeamish, don't look at the next slide, and I'll give you a warning when it's time. You can look back. So here's in medicine, we have rules, or in traditional medicine, we have rules. You know, you're over 35, you're a female, you should start getting x-rays of your breasts every so often. And personalized medicine wants to change all that. They want to use your genome to deliver a medicine that's appropriate to you, not necessarily to someone else. And this is a very good example. It turns out that if you have a particular set of genetic markers, for those of you that know this sort of thing, it's your HLA type. If you have a particular HLA type and you have seizures and you're given Tegretol for seizures, with almost certainty your skin will fall off. It's called Stevens-Johnson syndrome, and here's some images of people that have this condition. But it's completely preventable. If we knew your genome, we could see if you have these markers. You have these markers, we don't give you Tegretol. Problem solved. More generally, okay, now you can look back if the squeamish can look back on the slides here. You can use genetic markers to understand the causes of disease in order to work on treatments and cures. You can use genetic markers to diagnose a disease. You can use genetic markers to infer the propensity that you're going to get this disease, so you can try to take measures to prevent it. You can use genetic markers to predict a bad reaction to a drug, as I just showed you, and you can predict favorable responses to a drug, again, using your genomics. So there's all sorts of interesting applications here. And the immediate question is, well, how do you find these associations between these genetic markers, these SNPs, and various traits, like how tall you are, or are you going to get some disease? And there's been an approach in the genetics community for many, many years, which is rather tedious, and you certainly can't cover the whole genome. But with the advent of this genomics revolution and whole genomes that are available now, you can just do the obvious thing, which is called a genome-wide association study. And that obvious thing is to collect a set of individuals, measure some trait that you're interested in, or a set of traits that you're interested in, measure their genetic markers, do this whole genome sequencing, which only costs $1,000 a person now, and then do statistical tests looking for associations between the markers, the SNPs, and the trait. Okay, this has been a very successful endeavor. This graph shows each chromosome. Let me see if I can, yeah. So this bar here is chromosome 1, this bar here is chromosome 2. And then each colored dot represents some study, some GWAS that was done, and found a statistically significant, or one or more statistically significant associations between some trait and the location on the genome shown here. This slide is very old now. As of 2015, it would be just a mess. If you plot the number of associations that have been found as a function of time, it's growing exponentially. All right, so now that's the problem. Let's look at the data and consider both some statistical issues and computational issues around this data. So first of all, GWAS data are big. That's why I'm talking about it today. And one of the reasons for this is this notion that a small number of markers determine a trait. For example, in the Stevens-Johnson syndrome example that I gave you, or you've probably heard about sickle cell anemia, where one nucleotide change can determine whether you're going to have sickle cell anemia or not. Those are the exceptions, not the rule. The rule is, for most interesting traits, certainly traits involve common disease, these traits are highly polygenic. What that means is you have many, many SNPs influencing the trait, and the strength of influence for any given SNP is relatively small. And so an example of that is human height. There was a study done in Nature, 2010. They found hundreds of regions of the genome, or at least 100 regions of the genome, containing SNPs that were influencing the trait height. And because of this, you need huge sample sizes to get a comprehensive picture of how the genome influences a given trait. So there's groups now developing data sets with sample sizes in the hundreds of thousands and even the millions. And then to add to that, we have lots and lots of variants to consider. So when you have a data set with hundreds of thousands of individuals in it, you're going to see tens of millions of differences between individuals, as I was discussing before. So it's big in both directions. It has a large sample size, and it has lots of dimensions, these 10 million variants. And in fact, it's a big dimensional problem because the number of variants is typically greater than the number of, than the sample size. Okay, so not only is the data big, but it's confounded. This is a very simple example of confounding. Let's just consider a single SNP, and let's suppose it can have one of two values, A or T. And let's suppose we gather some data from two populations. Maybe there are two different ethnicities, or one's from Europe, one's from Africa. One of the populations is from Europe, or one of the populations is from Africa. And let's just suppose that in population 1, there's more A's than T's, and in population 2, there's more T's than A's, as shown here. And now we're going to do a GWAS, so we're going to identify a set of people from our two populations that have the disease, and we're going to identify a set of people from the population that don't have the disease. And let's just suppose in that process we end up with more people with the disease from population 1, and conversely we end up with more people without the disease from population 2. So in that case, if you just look at the data here, and you ignore the population, so over here you see, people with the disease, more A's than T's, people without the disease, more T's than A's. And you conclude that there's a correlation. But this correlation is not causal. It's not, at least it's not directly causal. Nowhere here did I assume that the SNP was actually, the SNP that you have, the value of the SNP, the A or the T, was influencing the trait. Okay, so in terms of graphical models, it looks like this. You have some hidden population variable. The population influences the value of the SNP, and the population influences the trait. There's no direct causal influence from the SNP to the trait, yet you find a correlation between the two. So in the genomics community, this is considered a spurious association. It's a correlation, but it's not directly causal. So it's a spurious correlation. Some people call it a false positive. Some people call it a type 1 error. Whatever, it's all the same thing. It's a mistake. Okay, so there's several ways to get this sort of confounding. Population structure is one of them. That's what I just showed you. And there's also family relatedness. And as your data sets grow in size, you tend to see more and more family relatedness. Certainly between distantly related people, you start to see more of that. And also the way studies are built, usually as they grow, there's an interest to get everybody in families, either nuclear families or extended families together. And you see more and more family relatedness in these data sets. And so when you go to big data, you see more and more confounding. All right, so now let's get into the nitty-gritty here. Here's a causal model for GWAS. We have a set of, these are all the SNPs lined up here. Some fraction of them will be causal, meaning there's, they're causal, and we're going to represent this with arcs from the SNPs directly to Y. Then you have a whole bunch of SNPs that aren't causal. Okay, no direct arcs here, but then you have the confounders. So this is, I show it as a single variable, but it could be multiple variables, hidden variables that influence all the SNPs and connect them all. So remember I was saying this is a very easy problem from a causal standpoint, and this sort of illustrates that. If there's an edge between a SNP and a trait, it's very clear what the direction of that edge is. It's the SNPs that are causing the trait. It's not the trait causing the SNPs. So really the only causal problem here is figuring out which of these SNPs have an arc to Y. That's the key question. So yes, from a causal standpoint, that's very simple. But we'll see when big data, when we get into the big data issues here, it's going to be more difficult than it sounds. Okay, so one thing I've done here is just reorder the SNPs now in the order along the genome, just to emphasize that these causal SNPs come from across the genome. They're not all bunched together. You might have one from chromosome 1, one from chromosome 5, one from chromosome 17, whatever. And another thing I'd like to mention is that this diagram that I just showed you is somewhat idealized. There are some other issues here. For example, due to recombination, this is the process where sperm cells and egg cells are created, where in the parent, 2 copies of the chromosome come together and you get this crossover effect, as illustrated here. It's really not important to go into the biology very much, but the upshot of this recombination is that SNPs that are close to each other, physically close on the genome, are highly correlated with each other, which I've tried to represent with these arcs here. Okay, so that's one issue. Another issue is that you can get a direct arc from the confounder to Y. An example of that is population. If the confounder is population, population is tightly correlated with environment, and you might have environmental effects that affect the trait as well. Okay, so those are two additional issues that go beyond this idealized graph, but for purposes of this discussion today, this is plenty complicated enough to illustrate lots of examples, and these other issues can be dealt with, and I'll actually come back to one of them near the end of the talk. So this is the causal model. I want to emphasize that the number of causal SNPs is large-ish, but not that big. You can have 100, for any given trait, you might have 100 SNPs influencing the trait, you might have 1,000, you might have 10,000. Fairly big, but not as big as the number of total SNPs, which is on the order of 10 million. At least that's what we're shooting for in the community. And then the sample size is also large, on the order of 1 million. Again, that's what we're shooting for. Nowadays, they're more like 10,000, but this is where the community is heading. And again, to make this talk a bit simpler, I'm going to just focus on Y, the trait being a continuous variable, and even more simplifications, assume that the relationship between Y and the causal SNPs are linear additive, which actually is a very good assumption for many different traits. The Mendel's P's example with dominance and recessive, that again is more of an exception than a rule. Okay, so in the genomics community, the frequentist approach is common. What they want to ask, is there a direct arc or direct edge from a particular SNP to the trait? They do a statistical test, for example, a likelihood ratio test, which yields a p-value. So the null hypothesis is that there is no edge between, say, S1 and Y. Compute a likelihood ratio for that and get a p-value. And the most important thing about this slide is that the genomics community is absolutely obsessed with controlling type 1 error. They do not like false positives. They do not like these spurious associations. And I'll emphasize avoiding type 1 error, or at least controlling type 1 error in the rest of the talk. Okay, so what was typically done until recently is obviously incorrect. People would do simple marginal tests looking for correlations between this, say, this SNP and the trait. So they compute the likelihood of Y given S1 and divide by probability of Y, get a likelihood ratio, do a likelihood ratio test. And of course, you're going to get type 1 error, and it's kind of obvious when you understand graphical models. So you have all these deconnected paths between the given SNP that you're testing. They go through H, then they go through a causal SNP, which you're not conditioning on, and they go to Y. And so now you have a correlation, and the correlation is spurious. There's no direct arc here, but you do see the correlation due to all these paths. All right, so what do we do? Well, the obvious thing to do is to condition on all the causal SNPs, and then you break all these paths, and you can then control type 1 error. And of course, the problem is you don't know which SNPs are causal. Okay, so there's two options that people have looked at. One is to condition on all the SNPs, except for the one you're testing. So if you're testing S1 and Y, condition on everything except S1. And another alternative that's been explored is just conditioning on some SNPs, use heuristic feature selection algorithm, try to find a lot of the SNPs that are candidates for being causal, condition on those when you're testing other SNPs, and then hope for the best. In either case, in either option, we're probably going to be conditioning on lots of SNPs. So how do we do that and keep the complexity of the model manageable? And when we do that, will we control type 1 error? So let's look at that. One approach that the community has looked at is very simple, Bayesian linear regression with a Gaussian prior, otherwise known as L2 regularization. This is what it looks like as a plate model. If you don't understand plate models, you can look over here. This is these are the distributions. So what you do is you model Y as a linear regression on the SNPs, which I'm just listing as X now because we don't know whether they're causal or not. And then for each relationship, you have a linear weight, beta 1 for this arc, beta k for this arc. You assume that the distributions on beta are mutually, or the beta is mutually independent, and that each beta has a normal distribution with some unknown variance that you can learn from the data. So when you take this model and integrate out beta, as I've shown here, you get this likelihood. And some of you might recognize this by a different name. This turns out to be a linear mixed model with a covariance or similarity matrix equal to XX transpose, where X again is are the SNPs. So basically, this matrix, the ijth element of this matrix, corresponds to the dot product of the SNPs for person I and person J. It basically says how similar the two people are. So it's a linear mixed model. It's also, if you allow X to grow, it's a Gaussian process with a linear covariance or kernel function. So the point here is this is a very simple idea. It's a very good idea. It's been invented many, many times. It has lots of different names. People know it by different names. But again, it's all exactly the same thing. All right, so let's try this very simple thing. We did this in an experiment, a large set of experiments that we've done. We just published this a few months ago. We generated lots and lots of different data sets. Some of the data sets used SNPs from real data, and then we generated the phenotype. Other data sets, we generated both the SNPs and the phenotype, and we tried to vary parameters as much as we can, how many causal SNPs you have, how strong the signals are from the SNPs to the trait. And then we tried these two different approaches, conditioning on all SNPs to see if we controlled type 1 error, and we also tried feature selection. And the type of feature selection that we used is marginal regression. I'll show you that in a moment. We also dabbled with forward-backward selection, which probably most of you have heard of, and the results are about the same. Okay, so here's the sort of feature selection we use. So the goal here is, before you actually do the GWAS and test each SNP to see whether it's correlated with the trait, you want to identify some putative causal SNPs so you can condition on them, avoid those paths that go through the causal SNPs, and avoid type 1 error. And so in this approach, we use trained test splits of the data. And for each partition of the data, we just do compute marginal p-values, the thing that we know doesn't work, that we know that has type 1 control problems, but type 1 error control issues. And we order the SNPs by these p-values, the smallest p-values, the strongest signals first, and then we build a model, one of these linear mixed models or Bayesian linear regressions, with using no SNPs, using the first SNP in the list, using the first 2 SNPs in the list, using the first 4 SNPs in the list, and so forth. And we build that up, and so we're just trying different numbers of SNPs, and we see which number yields the best prediction of the test data. Okay, very, very straightforward. This is rather crude, but with the size of the data we have, it's one of the few things that's feasible. So I'll show you the answer, and then I'll show you an example of one of the experiments we did. It turns out this feature selection approach does not work at all. You do not control type 1 error. However, and fortunately, this very simple procedure of using Bayesian linear regression does indeed control type 1 error very well. So let me show you an experiment. We did many experiments like this. This is just one example. Here we generated synthetic SNPs, 50,000 of them for 4,000 individuals. We generated family relatedness. We literally mated individuals in the computer, or maybe not so literally. Figuratively did that. And then we chose a subset of SNPs to be causal. We generated the traits from those with various amounts of signal. And the results you see here are a result of averaging over these different sets of particular experiments. So this is type 1 error. This is power. This is type 1 error as a function of the p-value cutoff that we use. If you're controlling type 1 error, you're going to lie along this diagonal. And there's some error bars here corresponding to confidence intervals under the assumption that you are controlling type 1 error. So linear regression, these are the marginal tests. They do horribly. Feature selection does better than linear regression. That's good. But you really need to condition on all the SNPs, again, except for the one you're testing, to get control of type 1 error. In terms of power, linear regression does poorly, and both the other methods do fairly well. Okay. Again, this is just a representative result, but so we did many, many experiments like that, and you can look at the paper if you want to get into the details. But so what's interesting here is that feature selection doesn't work. So feature selection picks many of the causal SNPs. If you go back and look, you see very successfully it picks over 90%, 99% of the causal SNPs. It identifies them. But when you have just 1 or 2 or 3 causal SNPs that are actually causal but not identified by this feature selection procedure, it creates these open paths. It does so for many, many SNPs, millions of SNPs, or in our experiments more like tens of thousands of SNPs. But it creates these open paths for many SNPs, and you see this horrible lack of control of type 1 error. Okay, and what's sort of interesting about this is that this model that we're using, L2 regularized Bayesian linear regression, it's not the right model. In that model, if you recall, we're assuming each linear weight comes from a normal distribution centered at 0 with some variance. So in effect, we're assuming each and every SNP is potentially causal. And we know, in fact, that only a relatively small subset of the SNPs are causal, at least in the experiments that we've done here, these synthetic experiments. So even though this model that we're using is fairly misspecified, it ends up controlling type 1 error very well and yielding good power. So we got a bit lucky there. Okay, so now that we have sort of looked at the statistical issues and we're fairly comfortable with that. Now let's look at the computational issues. So setting this in the context of Mike's talk, our philosophy, at least with this problem, was let's get the statistical issues right first and then worry about computation. And I'm not claiming that that always works, but in this particular case, we got a good result. The problem in this, with this genomics example is sample size. The problem with computation is sample size. So remember, we're shooting for sample sizes in the millions, and we're going to have to deal with SNPs in the tens of millions, number of SNPs in the tens of millions. And we like this linear mixed model or Bayesian linear regression. It works well from a statistical standpoint. But if we're going to evaluate this model against m SNPs, the runtime is OMN cubed. Thankfully, it's linear in m, but it's unthankfully, it's cubic in n. N is on the order of 1 million, completely not feasible. So what do we do? Okay, well, we got lucky. It turns out that algebra comes to the rescue here. It's a linear problem, and with some clever algebra, or really more straightforward algebra, you can get the runtime from OMN cubed down to OMN and this other factor k, and we'll get into that in a moment, but k is going to be less than n. So this is an algorithm called FASTLM. FAST stands for factored spectrally transformed, but wink wink, it actually stands for fast too. So there's basically two algebraic tricks involved in making FASTLM fast. The first is that if you do the algebra, if you do a spectral decomposition of the similarity matrix K, okay, and you take UT and you use UT to rotate all your data, rotate the phenotype, rotate the SNP data, and if you have any covariates, rotate them as well. You can turn the linear mixed model problem, this is the likelihood for the linear mixed model problem, into a linear regression. This is basically linear regression in a rather obtuse form, but it's linear regression nonetheless, and that reduces the runtime down to OMN squared. So that's one factor. The other trick, again algebraic, is to assume that you have k less than n SNPs that you're using to compute the similarity between individuals. And I'll come back to that issue in a moment, but make that assumption and use the fact that the similarity matrix is this product of the SNP data times itself, basically. And when you use these two things in combination, you can get the runtime down to OMNK, linear in all three. And some of you might be thinking, now wait a minute, you just told me you had 10 million SNPs, you wanted a condition on all of them, and your sample size is 1 million. So k is 10 million and n is 1 million, so k is not less than n. Well, we're going to use this feature that Mother Nature gave us, which is that SNPs that are very near each other are tightly correlated with each other due to this recombination effect. Okay, so the hope is that instead of conditioning on every SNP, you can condition on every other SNP or every third SNP or every fourth SNP and still capture enough of the conditioning signal so as to control type 1 error. We did this with mouse data where the coupling is very tight and it works beautifully. You can use either sample sizes on the order of 1,000 or 2,000, worked very well to control type 1 error. With humans, we don't know yet. No one's produced the data set that we need to determine exactly how many SNPs you can ignore, or conversely, how many SNPs you need to include in order to control type 1 error. So that's an open question, and we're on it, let's put it that way. So to sum up, in this genomics problem, the causal issue was fairly easy, but because you have high dimensionality, because you have confounding, because you have large samples, things can get tough. We happen to get lucky in this case. If you take this rather misspecified model using L2 Bayesian linear regression, otherwise known as a linear mixed model, otherwise known as a Gaussian process, if you use this model, it turns out you can control type 1 error very well in this very large problem. And again, we got lucky in the sense that when it came to the computational issues of sample size, algebra came to the rescue. So with that, I'd just like to thank my collaborators and thank you for listening.
We have time for questions. If you go to the mic, that would be helpful. Rich is on his way.
Quick one. Your model, and maybe in the real data, assumes single SNPs are the causal factors. How about interactions between them?
Sure. So we assumed that it was a linear additive model for this presentation, but that's not always the case. In fact, genomics researchers have a name for it. It's called epistasis, where the effect of two SNPs on a trait is not just the sum of each one. And yes, you can modify these models to incorporate those epistatic interactions.
What does it do to the computation?
Well, if you want to consider all pairs instantly, you're m squared. So there are some clever approaches that try to bound the strength of the signal based on univariate interactions and making various assumptions. We tested some of those models, and it turns out they miss about a good half to more of the associations than if you do with a brute force approach. So the technology is not there yet to really address that, other than cloud computing, if you're willing to heat up a whole lot of computers, because you can do all the testing in parallel.
Okay. Next question.
Can you say something? No. I have no idea how to turn this darn thing on, but can you say something more about the data that would be needed in order to control type 1 error in human studies?
Well, we don't know yet. There's several groups producing very large data sets. 23andMe has one. We can't get access to it right now, but they have 600,000 or 700,000 individuals in that data set. We're hoping to be able to gain access to that and do an experiment there. With that data, Kaiser has a couple 100,000 sample size now. Genomics England is working up a sample size on the order of 1 million. And once we have this data, we're going to try it. We're going to do it the hard way, conditioning on everything, get a right answer, and then try various subsets of the SNPs and see whether we can retain the type 1 control that we get in the full analysis. And of course, that's going to involve a lot of computation, but I'm fortunate enough to be at Microsoft where that's not too much of an issue.
We have time for maybe one or two more questions.
David Allison, University of Alabama at Birmingham. Very nice talk, thank you.
Thank you.
I was surprised to not hear you mention the ideas of conditioning on familial information as a way of really getting, in a sense, you might say, the right instruments to condition on.
Yes.
We and others have shown if you condition on the parent's mating type at the locus under study, that is a sufficient thing to condition on to remove all confounding except for physically linked loci. Now, that's not present, and that information is not present in every data set, but when you have it,
Absolutely.
you've got a perfect covariate.
If you have it and it's good data, we both know that people don't always tell the truth about who their parents are and vice versa. But if you have good data of that nature, then yes.
Okay, last question.
From UC Berkeley. So it seems like you end up doing feature selection because the computation saving, just like the marginal and forward didn't work. Would you agree? Because you end up doing the every other ones. You keep kind of the structure.
I didn't quite get the question. Can you repeat it?
You seem to have done some sort of feature selection because you were using every other SNPs.
Okay. So there's two very different kinds of feature selection. One is to use some heuristic method that actually looks at the phenotype and tries to figure out what the causal SNPs are, or a putative set of causal SNPs. That doesn't work. But if you just go along the genome and take the other kind of feature selection is to go along the genome and take every second SNP, every third SNP, every fourth SNP. In that way, using the tight correlations between adjacent SNPs as leverage for conditioning on the right SNPs, that does work. So, important distinction. Yes, thank you.